we're saying now is we're going to do a short recap. Now, I have my glomerular capillary. I have increased pressure. Which pressure? Increased hydrostatic pressure. And how is that? Because of the diameter differences. Larger, smaller. Now, the filtrate is going to come through the 3 layers. And that filtrate is called a glomerular filtrate. This is ultrafiltration, so you have lots of things. Within there, you have lots of water, lots of sodium, and lots of glucose. You also have other things like urea and so on. I want to focus on these 3 things. Okay. Now, this is ultrafiltration. Glucose should never be in the urine. Eventually, everything here goes to the urine. We don't want the glucose, so we need to, you can't say absorb it, that will give you wrong mark. Reabsorb. Very important, reabsorb glucose somehow. Maybe the body needs more water. Okay, so we're going to reabsorb water. Maybe the body needs a bit more sodium. We need to reabsorb sodium. 85% of reabsorption occurs at the proximal convoluted tubule. And this is why your blood vessel here is following this PCT, because it wants to reabsorb. So we need to know, how does it reabsorb the glucose in here? Exam focus for PCT, how does it reabsorb glucose? Glucose should never be in the urine. If it is, then probably diabetic. So I want to draw the PCT in here with the blood vessel. So what we have is my PCT. Obviously it's convoluted, but I'm going to do it in a way it's not, so you can understand. This is my convoluted tubule. So and this is the lumen of the tubule. This is the lumen. So this tubule here, okay, here, this tubule, this part here, I've drawn it here. So that line is made from cells. Which cells? Endothelial or epithelial? Epithelial. Endo is only for blood vessel. So these are the epithelial cells lining the tubule. I have one epithelial cells here and many cells here. I'm drawing the outer part. And that's the lumen, that's the space. So what do I have inside here? I have the filtrate. So I have lots of sodium, right? And I have water. I also have glucose. Remember, our focus here is to take the glucose inside. So let's see how that works. And then we have our blood vessel. This is the blood vessel here. I'm going to draw it here, right next to it. So this is my blood vessel. So what happens first is sodium is in the epithelial cell. There are 3 places here, 3 places. The first place is your blood vessel, number 1. Second place is the epithelial cells of the PCT. The third place is the lumen of the PCT. Lumen is the space, not the hollow space. Yep. So what happens first is sodium is actively pumped into the blood vessel. So that requires ATP. So epithelial cell has lots of mitochondria as an adaptive feature, and it's got a large surface area as well to allow it to pump sodium. So sodium is pumped out from here to here by active transport. Number 2, what happens, because sodium now is from here is going really, really low, okay? In the lumen, remember we have sodium. That sodium was from the filtration, ultrafiltration. The sodium is higher here. We have made a gradient. Sodium is high in the lumen, and in the epithelial cell is low. There's a gradient. There is a protein. Through that protein, sodium is going to go from the lumen into the epithelial cell down the gradient. No ATP required. Okay? And glucose will use the opportunity to use the same protein. So we call that co-transport. So remember, our focus here is glucose. So glucose has used sodium's channel as a co-transport. Now glucose is here. Then the glucose from the epithelial cells of the PCT will go to the blood vessel via diffusion. So that's how glucose gets reabsorbed. So you need to know the adaptive feature for PCT and how glucose gets reabsorbed. And